Hi, my name is Jessica Martin, and today I am going to be going over my digestive system portfolio assignment. The portion that I decided to focus on was the role of the liver and pancreas in regulating digestion. So we'll go ahead and get started in taking a look at that. So this first slide is just an overview of the entire digestive system. So up here we can see that it starts in the mouth and that we go down the esophagus into the stomach, into the small intestine here. This first portion is called the duodenum. The ileum, which is the next portion. And then we go into the large intestine, starting with the cecum, ending with the colon and the rectum, and out the anus. So this is the whole picture, but we're specifically going to be looking at the secretions of the liver and the pancreas, as I mentioned before, which are going to enter the digestive system through the duodenum. So that's important to note. The first one that we're going to talk about is the pancreas. The pancreas is considered to be both an exocrine and an endocrine gland. So when we're talking about the pancreas as an endocrine gland, we are typically talking about the secretion of insulin and glucagon and their role in regulating the blood glucose level. However, today we are talking more about the exocrine function of the pancreas, and in the exocrine tissue of the pancreas, they form structures called acinis, which are these little circular duct-like shapes here. And they will create the pancreatic secretions, which will then enter the center of them and then go out the ducts. These ducts will all combine into the pancreatic duct, which will then carry the pancreatic secretions to the duodenum. They are responsible for secreting both digestive enzymes as well as a sodium bicarbonate solution. So first we'll talk a little bit about pancreatic enzymes. They are released when the small intestine is distended, when food is present in the intestine and in response to neural signals and the hormone CCK. Most pancreatic enzymes are secreted in an inactive form called a zymogen. They must be activated first in order to have an effect. So typically what that would look like, as you can kind of see in this, it's a cascade, so a chain of events. And it will typically start with enteropeptidase, which will turn trypsinogen, which is a zymogen, into trypsin, which is the active form. And then the role of trypsin is to go through and convert all of the other zymogens into their active forms. So for example, we would turn chymotrypsinogen into chymotrypsin, prolatase into elastase, kallikreinogen into kallikrein, and procarboxyl peptidase into carboxypeptidase. So that is how that would function. And again, all of these would be secreted through the pancreatic duct into the duodenum. As I mentioned, the pancreas is also responsible for the secretion of bicarbonate. Bicarbonate is important because it's released into the duodenum to neutralize the chyme that comes from the stomach. The stomach is a much more acidic environment than the small intestine, and so once the chyme enters the small intestine, it's really important that we do neutralize that. So the cells that are responsible for secreting the bicarbonate have a large amount of carbonic anhydrase, which is the enzyme that is responsible for combining carbon dioxide and water to form the bicarbonate. The formed bicarbonate is then put into the lumen of the digestive system, or into the lumen of the gland here. And as you can see, it is an exchange protein which brings chloride back into the cell. So the bicarbonate leaves, the chloride comes in. The chloride ion, though, does not stay inside the cell. As you can see, there's this channel that brings it back out into the lumen. And because there's this constant flow of negative ions between the bicarbonate and the chloride ion into the lumen, that attracts positive ions like sodium down their concentration gradient into the lumen as well. And because of the movement of both the bicarbonate and the sodium into the lumen, we end up with a watery sodium bicarbonate solution, which is what ends up being our buffer in the stomach or in the small intestine. Next, we'll go ahead and dive a little deeper into the liver. So the liver is really notable for secreting bile. The bile does not go straight from the liver into the duodenum. So the liver, hepatocytes or liver cells are the ones that produce the bile, but then it is collected in the gallbladder, which is represented by this little green structure here. And once it's ready to be released into the duodenum, it would go through the common bile duct and out to the duodenum to do what it needs to do. Bile is really notable in its role in helping the digestion of lipids. So bile is composed of bile salts, bile pigments, which are waste products like bilirubin, as well as cholesterol. And like I mentioned before, they're really important in the breakdown of lipids. So kind of what that looks like is that lipids in chyme are hydrophobic or water hating. So they bind together in large clumps. This is where the bile salts come in. Bile salts are polar, kind of like the phospholipids in a cell membrane. They have a hydrophobic and a hydrophilic end, so they will surround little droplets. So they'll take the big droplets and turn them into little droplets called emulsions, which then allow the lipids to be digested by lipases. So that would turn our larger tri or diglycerides into monoglycerides or free fatty acids, which can then form micelles, which then would allow the active or passive absorption of the lipid into the body. So it's really important here. Once the bile salts are done and they've done their job, then they are reabsorbed into the body for reuse. However, I did mention that bile had a couple other components, the bile pigments and the cholesterol. Neither of those are reabsorbed. So those will go on to be excreted in the feces as they are waste products and are to be excreted from the body. So that is the end of the content that I have. I did put together this worksheet for my peers to go over and kind of review the content that I went over about the lipid or the pancreas and the liver. So the questions that I have are: what organ secretes a sodium bicarbonate solution? Why is this important? The second question: what does bile help to digest? Third question: is the pancreas an endocrine or exocrine gland? Fourth question is: where did the liver and pancreas secrete their products into the digestive tract? And fifth question is: what must occur for pancreatic enzymes to be activated? So those are just a little bit of food for thought. I will attach the answers to these questions along with the presentation slides, but they should all be answerable with the video. And I hope that it has been informative. Other than that, thank you.